Hello, good morning, everyone. I hope you guys had a restful engagement week last week and you're a little bit recharged. Today, we will be taking up the Test 1 review package. If you have not already downloaded a copy, you can see a link in the chat box that will take you directly to the course page, and you can download a copy and do it together. If you prefer to do it later, that's okay as well. You can just watch and copy down the stuff in your notebook. That will work as well. The test, of course, is on Wednesday, and the details are posted on eCentennial. There's also a practice quiz, right? Self-quiz that you can do. I got several emails asking me to increase the number of attempts you can have for the self-quiz. So I bumped that up from 3 attempts to 5 attempts. But I wouldn't go, any more than 5, just because, it's not the best way to spend your time, right? Like, you want to do it maybe like a few times, and then if you didn't do too well, go study a little bit, and then just try it a few more times, and then just leave it at that, right? Also, I got questions asking me if the review package are the only thing you have to study for to prepare for the test. Not so. You also have to look at the PowerPoint slides, as well as all the materials that we've covered before in the previous tutorials, as well as previous study guides. Okay, so this is not an exhaustive list of everything you have to know, but it's a good place to start. So let's get started. First question, lecture 2, macromolecule. What holds the atoms together in a water molecule? How would you describe a water molecule? What interactions exist between two adjacent water molecules? Okay, so a couple of things here that you should know. Water molecule is going to be a molecular compound. Okay, so water, I'll say H2O. H2O is a molecular compound. Okay, so you can draw out water. There's 1 oxygen here, 2 hydrogen. And anybody know what is the name of the bond that holds them together? What do we call this bond that holds the oxygen and the hydrogen together? Anyone?
Covalent bond.
That's correct. That is the covalent bond. And a covalent bond is a bond where the electrons are being shared. Okay, so they're sharing of electrons, as opposed to a transfer of electron, right? In the case of an ionic bond. Okay, so we talk about how even though they're sharing the electrons, but it's not necessarily going to be an even distribution of the electron, because oxygen has a higher attraction for electron. It's going to be pulling the electrons towards the cell, and as such, the oxygen atom is going to be slightly negative, whereas the hydrogens are going to be slightly positive. So how would I describe the water molecule? The water molecule is going to be a polar molecule. Okay, polar. So that means one part is slightly negative, while another part is slightly positive. Okay. However, it's very important to know that this uneven distribution of electrons cause it to be polar, yet the entire molecule is still going to be neutral. Okay. Neutral. Okay. No excess or deficit of electrons. Okay, so I would say the water molecule is polar as well as neutral. What interactions exist between two adjacent water molecules? We also talk about that. If I have one H2O here, and then I put another H2O nearby, the slightly negative oxygen will be attracted to the slightly positive hydrogen of another water molecule. And then there, you have what we call a hydrogen bond. Hydrogen bond. Okay. That's all. That's it for the first question. Next one, if you supply radioactive nitrogen atoms, something that can be traced and detected, to a cell and let it grow, what molecules will you expect to find the radioactive labels in? In other words, right, where will all these nitrogen goes within the cell? Okay, so you got this question is about macromolecules, of course. So let's write down the macromolecules. We have carbohydrates. Okay, so you think about what kind of atom makes up the carbohydrates, right? We have hydrogen, carbon, and oxygen. Okay. Can talk about proteins. Can someone tell me what are the major atoms that are found in proteins? You can type it out. You can unmute. Proteins? What's in proteins? Yeah, Tyler said CHO and N. That's very good. Okay. Also trace of sulfur, right? Yeah, that's important to remember. Yes, thank you. And sometimes S. That's correct. Lipids. Yeah, what do we have in lipids? Lipids, we have C, right? H and O as well. And then for nucleic acid, we have CHO N, right, as well as phosphorus. Right? So if you feed the cell with radioactive nitrogen, it is going to show up in the nucleic acid, and it's going to show up in the proteins. Okay, so this is just like rather than asking you straight up, right, like which macromolecules has nitrogen, like basically we just put it into like an application setting, right? All right, a lot of stuff going on here for question number 3. Regarding DNA, what is the monomer of DNA? So the monomer of DNA is going to be nucleic acid. Oh, sorry, nucleotide, I mean nucleotide. Okay, so the nucleotide has different parts to it, right? There is the deoxyribose, which is a sugar missing an oxygen. Ribose is the sugar, but it's missing oxygen. So we say deoxyribose. Sorry, I can't spell this morning. Deoxyribose. We are also going to have a phosphate group. Phosphate group. Okay, in chemistry, you learn phosphate group. It's like PO4, right? 3 minus. And then there is also going to be a nitrogen base. Nitrogen base. Okay. So for DNA, the nitrogen base could be A, C, G, or T. Right? Adenine, cytosine, guanine, or thymine. That's part A. Part B. How are nitrogenous base or nitrogen base, how are they classified? Anybody remember? What are the two groups of nitrogenous base? What do we classify them as? Yeah, they could be purines, right, or pyrimidines. Purines or pyrimidines. Okay, so purines include the adenine and guanine. Right? And purine, if you remember, they are actually going to have 2 rings. 2 rings in their structure, right? 2 rings in their structure. There we go. 2 rings. I try to remember, like purine has 2 syllables, so 2 rings. And the pyrimidines include the cytosine, thymine, and thymine. Of course, it includes the uracil as well, but then we're talking about DNA in this particular question, so we won't have that. And for the cytosine and the thymine, for the pyrimidines, there is only going to be 1 ring, okay, to the structure. Something like that. Okay. So that's a one ring. What is the base pairing rule for DNA? Okay, so the base pairing rule is A pairs with T and C pairs with G. And I'm just going to skip over to E. What is the significance of base pairing rule? Why does A always goes with T and C always goes with G? Well, it actually has to do with the rings, right? Okay, so if you take a look at adenine, adenine has 2 rings. And when it pairs with thymine, the width of the DNA, it's going to be 3 rings across, right? And the same thing can be said for guanine and cytosine. 2 rings for guanine, 1 ring for cytosine. And this base pairing rule essentially, like if you look at the DNA, if you unwind the double helix, you will essentially always have 3 rings across. Okay, so like 1 ring, 2 rings, and then 3rd ring, right? 1 ring, 2 ring, 3 rings. Okay, because a purine will always pair with a pyrimidine, and it basically allows you to maintain a constant width along the length of the DNA molecules. Okay, so let me try to write that out for you. Okay, so maybe that was not the best diagram, but basically if you go across, right, it's going to be like 3 rings, right? 3 rings across. Okay. So that was E, right? Pairing a purine with a pyrimidine allows the DNA molecule to maintain a constant width lengthwise, we can say. Okay, so along the length of the DNA, it's always the same amount from one side to the other. Okay. Okay, what force holds the nitrogen base together along the length of the DNA molecule? So in other words, you know, if I have the A, C, G, T here, and then the other side would be T, G, C, A. What holds them together? Anybody remember? What is the force that holds them together? Okay, thank you, Tyler, again. That is the hydrogen bond. All right. 2 hydrogen bonds between A and T. 2 hydrogen bonds between A's and T. And 3 hydrogen bonds between C and G. Okay. So it's easier to pull it apart if you have a lot of A and T's compared to a lot of C and G. Any other question? Should probably worth mentioning at this point that the backbone, the DNA backbone right here, right? That is a sugar phosphate backbone. Sugar phosphate backbone. It's made up of the sugar and the phosphate, and then sort of the rugs of the DNA, that of the leather, that is made up of the nitrogenous base. All right. Moving on. Next question. What is dehydration synthesis? Illustrate with a diagram of this process. So that's quite easy, right? You have the monomers here, right? Monomers. And then in the process of dehydration, synthesis, you will connect the monomers together. Okay? Every time you connect the monomer together, you will generate some water. Okay. And of course, the opposite, to break down the polymer into the monomers, it would be hydrolysis. Hydrolysis, and you would need to have an addition of water molecule. Okay. So one of the questions that you saw before is something like this, right? Like if I have some kind of ring structure, right? All these corners is actually a carbon, and then there's an OH here. If you combine it with, let's say, another thing like that, right? And then I would ask you, what does it become? Well, you will be able to take an OH from one and an H from the other, and then in the end, you will create a new bond, right? Plus H2O. Okay, so the water that you took from here comes out, and then you essentially create a new connection between this carbon and that oxygen. Okay. Do you see that? So this structure is here, and then the pentagon along with the oxygen, that's from this molecule. Right? So you might remember we did this a while back, right? When we had a tutorial in lecture 2. Okay. Next question regarding enzymes. Okay. So what is an enzyme? Enzymes are proteins, right, that catalyze chemical reactions in the body. Okay, catalyze means they speed up chemical reactions, right, in the body. So everything that occurs in the body, in terms of reactions, we would need to have an enzyme for it. So how does an enzyme actually work? We talked about this in a lecture, I think, but it's worth mentioning it again in case you bump into a question like that. So over here we have energy, right? Energy, and then here we have time. Okay. So normally, let's say your reactant has this much energy. And this is your reactant. Reactant is the ingredients in a chemical reaction, right? So that's the reactant. It has this much energy, and in order for them to react, you will need to supply a little bit of energy, and then it would go ahead and react, and then the products will be down here with less energy. That's one type of reaction anyways, okay? So this energy that you need to supply, right, the amount of energy that you need to supply before the reaction can take place, this is called activation energy. Right? Activation energy, which is the amount of energy required to start a reaction. So to relate this with what we learned in the metabolism unit lesson, in glycolysis, right? In order for the glucose to split into the 2-pyruvate, you must supply 2 ATPs, right? To prime the molecule, right? To make it unstable, to make it willing to react, right? So that's the activation energy. So typically the activation energy is quite high for a lot of the reactions that we carry out in the body. And it would be going at a very slow rate if we do not have an enzyme. So in the presence of the enzyme, right, the enzyme allows you to lower the activation energy, okay, and still carry out the same reaction. Okay, so over here, this red curve, enzyme, how does it work, right? So enzyme lowers the activation energy. Okay, that's it. What factors affect the functionality of an enzyme? Of course, enzyme is a protein, so any factor that affects the shape of a protein is going to affect the functionality of the enzyme, right? Okay. Enzyme, of course, has a key and lock relationship with its target, with its substrate. So if you change the shape of the enzyme, you are going to affect how it's going to work. So go ahead, please help me out here. What are some factors that affects the shape of a protein? Excuse me. Anyone else wants to participate? What are some factors? What if this is a multi-select question, right? Select everything that would affect
Temperature.
temperature, definitely. Okay. And more importantly, you should think about how it's going to affect it, right? So temperature will, for example, start breaking down the hydrogen bonds, right? Which are what's holding the secondary structure together. Okay, so what else besides temperature? Just type it in the chat box if you want. Okay, yeah, pH, definitely. Okay, pH becomes more acidic, more basic. That will also disrupt the hydrogen bond and even the ionic bonds as well. Salt concentration, very good. Okay, and then I'm looking for one more, perhaps. Anybody? Yeah, exactly. The type of solution, right? So type of solution, for example, if it's a hydrophobic solution, it would cause the enzyme to flip inside out because normally, right, the enzyme in an aqueous environment, in the water-friendly environment, would keep all the hydrophobic parts on the inside. So it's not going to be, interacting with the hydrophilic environment. But if you switch the solution, then it will, go inside out, and then it's not going to work anymore. All right. What does the enzyme lactase do? All right, so lactase, anything that's ASE means it's probably an enzyme. So lactase breaks down lactose. Right? So lactose, and then the reaction with lactase is going to break it down into glucose plus galactose. Okay. Remember, right? Like people who don't have lactase, for example, they will not be able to break down lactose, and as such, they will become lactose intolerant. All right, moving on. Complete the following chart. This is a good chart to have, like with you, as just a quick reference, and you definitely should make your own and add, as much detail as you need to. But let's just keep it simple here. Carbohydrates, the monomer for carbohydrates are basically monosaccharides, right? Monosaccharides. Let me zoom in a little bit. And the types of monosaccharides we talked about in the class include glucose. Okay. There is also fructose and galactose. Okay. So these are the monosaccharides that we have. In terms of biological function for carbohydrates in general, source of energy, right? Okay. Example, cellular respiration. Okay. Another biological function is storage of energy. Okay. Such as starch and glycogen. Right? Starch is storage form in plants, of course, and glycogen is storage form in animals. We store them in the liver and we store them in the muscle. And one last biological function, structural support. Structural support, such as chitin for exoskeleton of like bugs and crustacean, like lobster, crabs. And another example of structural support would be cellulose. Okay, cellulose. I guess here we can say glucose, right? Glucose in cellular respiration. Examples. Well, we just listed a bunch, right? So whatever, we just copy a few. Starch, glycogen, okay, cellulose, dot, 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 dot. Okay. Next one is proteins. Proteins. Monomers of proteins, anybody remember? What is the monomer? What are the monomers for proteins? The amino acid is correct. Amino acids, right? There are 20 of them in total. 12 of them are non-essential. 8 of them are essential. Essential means your body cannot make it and you must eat them. It's probably a good idea to recognize what a amino acid looks like. You're not going to have to draw it, but you should recognize it. If I show you a bunch of pictures and I ask you which of the following is the general shape of a amino acid, you should be able to tell me that it is this guy. So again, there is the amino group here, the carboxylic group, and then there is the side chain. Okay. Amino group. Carboxylic group. Carboxylic acid, actually. And then this is the side chain. Okay, now listen carefully. And don't say I did not tell you. When you want to join 2 amino acids together, we create a peptide bond, right? A peptide bond. So if I asked you on the test, or if you see a question that says, what makes up a peptide bond? Right? What makes up a peptide? Actually, anybody want to tell me what makes up a peptide bond based on everything I told you right now? What is the best way to describe what a peptide bond is? By the way, okay, just so that you don't feel like these are like random questions that I pull out. These questions are like based on the test bank that I created, right? So I generate like a test for myself using the bank. So it would be similar to what you would see. And then I create these questions based on that, right? Okay, so these are very likely questions that you're going to see. So once again, my question is that if I ask you to describe what a peptide bond is, how would you answer? Okay, I'll do it. Where should I put it though? I'll put it up here. Okay. A peptide bond is or consists of an amino group connecting to a carboxylic acid, right? So in lecture 2, we did it, right? I think we did it. right here, right? You see the peptide bond? When you take the carboxylic acid and you connect it with the amino group, you create a peptide bond, and it looks something like this, right? Okay, right here. That's the peptide bond, right there. Understand? Okay. Carboxylic acid and the amino group. Okay. Biological functions. Many things. Okay. Help me out here. Okay. Type it in the chat box. There must be like 5, 6 different types of functions that we learned, you know, in the course so far. Yep. So catalyzed reactions, right? As enzymes, good. So enzyme would be an example, right? Okay, so I'll just put the example here. Muscular structure, hormones. Yep. So cell signaling or signaling, we'll say signaling. Signaling, and that would be hormones. Right? And it's always good to know a few hormones. So example, insulin. That's the one we keep on saying. Structure, yeah. So muscle. I don't know if I told you or not, but it's actin and myosin. Probably not, but in case you want to know. What's another one? Maintains pH. Yeah, could be. Not a big one that we talked about, but that could happen if we have an enzyme that does that. Structure, fuel, yeah, energy source. Okay. Other structure would be like, nails, hair. Okay, so this would be amino acids and peptide, Short's peptide can be fed into glycolysis and Krebs cycle. I don't think we talk about this in the lecture, but it's actually true. Depending on which amino acid it is, you can actually convert it into, the different intermediates within glycolysis or the intermediates in the Krebs cycle, and then you would metabolize them from that point. Georgia says skin. Yep, skin also has protein, right? The keratin. That is correct. Very good. Moving on. What do we have next? Lipids. Okay. The monomer, it's not really monomers, right? I keep on saying the lipids are kind of like components, but we have things like, glycerol, we have fatty acid, right? And then maybe phosphate group, right? Phosphate group, depending on what kind of lipids you're talking about. If we're just talking about triglyceride, like fat, then it would just be glycerol and fatty acid. If you're talking about the phospholipids, then you might have a phosphate group as well. Okay. So what is the function of lipids? Membrane component, right? Let me just create more space here. There we go. Oh, this is not going to make any sense for anyone who just download this later. Okay, membrane components, right? What are the major lipid membrane components? Well, there's the phospholipid plus cholesterol. Okay, lipids also will help in signaling. So such as hormones again. Okay. Remember, there's steroid hormones, and then there are protein hormones, right? Another one, so like example would be like estrogen. Okay. Another example would be insulation, right? And protection. Where you have some fat around major organs, keep them warm, protect them, give them a little bit of cushioning, right? And of course, fat is excellent storage form of energy. Okay. Next, we have nucleic acid. The monomer of nucleic acid should be clear by now. It is the nucleotide. Which again has the sugar, the phosphate, and the nitrogenous base, right? Okay. So just to list them out for completion. DNA, we have A, C, T, G. RNA, which is single-stranded, has A, C, U, and G. Main function, let's talk about DNA first. DNA, the main function would be providing instructions to make proteins. Okay. It is the genetic information that is inherited to the next generation, right? The blueprint of life, right? They say. And then RNA, well, you know, RNA has a lot of functions. We only learned one type of RNA so far. So messenger RNA is used by ribosomes to make proteins. Okay. There are other types of RNA that we will learn later on, and so far, example would be limited to DNA and RNA. There we go. Okay. Remember, feel free to ask questions if you have any. Number 7, how is the shape and structure of protein stabilized? Okay, so this is quite important to know the various structures of protein, the different levels and how they are stabilized. So for example, in the primary structure, right? We have the sequence of amino acid. That's what the primary structure means. Okay, primary means the sequence of amino acid, right? And they are held together by peptide bond. Okay, so that's a peptide bond, that's a peptide bond, right? Each peptide bond consists of the carboxylic acid of one amino acid connected to the amino group of another amino acid. So these balls, right? These are amino acids, and that here, that is the peptide bond. Okay, when you heat up a protein, you unfold the proteins, but the heat will not be able to break the peptide bond. Okay, so primary sequence is not affected by things like change in temperature. Okay, it's not affected by things like change in pH. To break this bond, you know, it actually requires an enzyme to cut it, for example. Okay, so secondary, this is the alpha helix or beta pleated sheets. Right? And what holds them together? They are stabilized by hydrogen bonds. And if you're not sure what hydrogen bond is, it basically means when a slightly negative portion of one molecule is attracted to the slightly negative portion of another molecule. Except in this case, the protein is a really, really big molecule, right? So you can have a slightly negative portion of the protein attracting to the slightly negative portion of the same protein in this case. Okay, so those interactions will help create these alpha helix as well as the beta pleated sheets. Okay, so stabilized by hydrogen bonds formed from the peptide backbone. Okay, and just to, I'll come back to this in a second, just to help you refresh your memory what a peptide backbone is. We talked about this in lecture 2, right? You see? Right, this whole thing, it just keeps on repeating itself, right? As you form the polypeptide, the backbone does not change, right? So you would have the hydrogen bonds of one part of the backbone interacting with the another part of the backbone. Okay, what changes is the side chains, right? Okay, so over here, stabilized by hydrogen bonds formed from the peptide backbone. So again, right, this primary structure that you have, it's now going to fold up. Right? Some parts going to have the helix, some parts going to have the zigzag, right? The plated sheets. Okay. These can easily be disrupted with heat. Hydrogen bond is very weak. You can crank up the temperature and then it will start to melt apart. A tertiary structure, right? These are interactions of side chains. Okay, and it creates the 3D structure, 3D shape, 3D conformation of the protein. Okay, so what holds them together? There's ionic interaction, right? Let's say you have a side chain with a Na plus or something, and then there's something Cl minus, and then they could form an ionic interaction, right? Okay. There is hydrophobic interaction. Things that are hydrophobic tends to clump together. There are other things like a disulfur bridge. Remember the traces of sulfur? So if you have one sulfur in one part and then another sulfur in another part, they actually can form a bridge to stabilize the structure. Okay, so on and so forth. Right. This is of course in the in one of the slides in the PowerPoint. And again, most proteins will stop at this structure, right? The tertiary. But some will continue to fold and form the quaternary structures. Okay, that's when you have multiple polypeptides coming together to form a protein complex. Okay, so this would be like, you know, peptide number 1, right? Peptide number 2. Then peptide number 3, and then peptide number 4. Each of these are individual polypeptides. Okay. Polypeptide. Okay, there you have it. And these are stabilized by the interaction between the polypeptides themselves. Okay. Moving on. Oh, that's it for lecture 2. Any questions in lecture 2 at all? Anything I missed? Any other concepts you want me to cover before we move on to number 3? Everybody still with me? Yeah. Great. Okay, list the major components of the cell membrane. We did this before, right? Let's just quickly do it again. We have the phospholipids, right? Phospholipids. Like that. Okay, so we have the hydrophilic head facing the aqueous outside or the inside. And then we have the hydrophobic tails, right? Which forms the major barrier in the middle. Okay, so because of this arrangement, things that are charged, things that are hydrophilic, or if they're big, they will not be able to cross easily. Okay, so things that are big, or if they're charged or polar, will not be able to cross the membrane by themselves. Okay, they're going to need some help to do it. Right, so to help them go through, what are we going to use to help these things pass through? What is in the membrane that helps us move these charged large polar molecules across? That's right. We're going to have transport proteins or membrane proteins, and we can have two types of membrane proteins. We can have the channel proteins. Okay, the channel protein is an example of an integral proteins, right? It goes all the way through. And so here we have the channel protein. Okay, so things could just like if this is charged, right? Let's put some positive around it. It won't be able to go through the membrane, so it's going to go through this channel. Okay, the channel is going to create a nice hydrophilic tunnel in the middle that allows a charged particle to go through. That's not the only one, right? You can also have the Pac-Man, right? Okay. The Pac-Man is a carrier protein. Okay, more on that later, but both of these are integral proteins, right? Okay, they also help stabilize the structure of the membrane by having these things in it. So if you crank up the heat, right, in the lab, then the phospholipids start to move further and further away from each other, and then the dye, the pigment rather, right, it's gonna seep out right from here. But then when you keep on heating it up, the proteins start to break down, and once the entire protein is broken down, then now you have like this gaping hole, right, and all the pigment just kind of come out, right? So that's why you see that sharp rise in absorbance in the lab. Okay, let me put the Pac-Man back in like that. Okay, what else is there? There are also these cholesterol right here and there. The more cholesterol you have, the more fluid your membrane is going to be. So these are cholesterol molecules. We might also have some, peripheral protein. Okay, the difference between integral and peripheral is integral goes all the way through. Peripheral is just kind of sticking to the side. You might have some on the outside, right? Do I have a question that talks about the functions later on? I don't think so. So let's just review that as well here, right? So if it's a peripheral proteins, they can do like, enzyme reaction, right? Enzymatic activity. They could do like signaling, right? All those 6 different functions that we talked about, right? It could be attachment to cytoskeleton, right? These are cytoskeleton, right? So all these things. You might have some another protein. Let me just draw another protein here. If I could find a space, right? You might have another protein here. Okay, and sometimes the proteins are gonna have sugar on them, right? So that creates what we call like a glycoprotein. That's important for cell recognition, right? Your white cell is gonna have to be able to tell apart what belongs to the body and what does not, right? We also have the glycolipid right here. Glycolipid. Like that, right? Same function. Any question? Good. Next one. Describe the central dogma, the central theme of biology. Okay, so basically we start with the, oops, sorry, give me a second. We start with the DNA in the nucleus. That's gonna be copied in the process of transcription. Okay, transcription. And then the RNA is going to now go to the cytoplasm, where it is going to be used by ribosomes to make proteins in the process of translation. All right. So there is your cell. There's your nucleus. Okay. So the transcription happens in the nucleus. We actually create it in the form of the messenger RNA. Okay, so you have your double helix DNA right here. And then the messenger RNA, of course, is going to be single strand. And that will exit into the cytoplasm. Right? Remember, the nucleus is surrounded by the nuclear envelope that has a lot of pores on it, right? Nuclear pores. So the mRNA is just going to exit through those pores into the cytoplasm. Just label it for you. Nucleus. This will be cytoplasm. And in the cytoplasm, we are going to have the ribosome. So the ribosome has two subunits, the big one and the small one, and they will clamp onto this messenger RNA. They will read the codes, and then it will make the polypeptide for you. Okay, it will make the polypeptide. So that is a protein. Okay, so that's the central dogma of biology. Be sure to mention the related cell structure and location. Okay, we did already. Okay. Next one. One of the functions of liver is to remove toxins from blood. Based on this function, which two organelles would you expect to be abundant in liver? We talked about this like a couple times already, right? So you would expect to have a lot of lysosome for sure. Okay, they contains a mixture of enzymes for breaking down macromolecules, right? Toxins, old organelles, right? And even bacteria if it's a white cell or something, right? What's another organelle that you would expect to have an abundance of? Yeah, Tyler and Hashana said smooth ER. That is definitely correct. Smooth ER. Right. That is involved in synthesis of lipids as well as detoxification, right? Detoxification. And you might also have a bit of peroxism as well. Peroxisms. Okay, so uses oxidation reaction, which is reaction that now you know what oxidation means, right? It means it rips electrons away from something. So it uses oxidation reactions to break down toxins and other molecules. Okay, generates H2O2 as a byproduct. Okay, and that of course is dangerous. It needs to be, broken down immediately before it causes any cell damage. Okay. I know it only asks for 2, but then, just want to throw in peroxisome there in case you see them. Describe the structure of mitochondria and state the function of each part. Okay, so this is of course very important after we learn about cellular respiration, right? So let's just see if we remember everything. Okay, so we have the outer membrane, of course. We have the inner membrane as well. That forms cristae, right? The finger-like projections, the folding. And we need to state the function of each part. Well, the outer membrane and the inner membrane basically creates a separation of the matrix from the intermembrane space, right? Now, this separation is important because, as you know, in electron transport chain, all these proteins, all these membrane proteins, right? They will be pumping hydrogen into the intermembrane space. Okay, so by having a double membrane system, you allow the hydrogen concentration to build up here. Okay, so allows H+ ion to build up. In here. And by building it up, you create a concentration difference, right? Which would then allow them to come back down into the matrix, right? So that concentration difference is what drives the production of ATP, right? Okay. So these purple things, those are the electron transport chain embedded within the cristae. Okay. What is going on in the matrix? Well, tons of stuff happens in the matrix. We have the pyruvate oxidation. And we also have the Krebs cycle. Okay. Maybe just one more note here. ATPase produce ATP, right? Using the H+ gradient. All right. Maybe another note here, inner membrane. The inner membrane prevents H+ from leaking through, right? We do not want the H+ to leak down. Remember, we had a question in the lecture 5 tutorial, and I asked you what happens if the inner membrane has a lot of holes in it, right? Well, if it has a lot of holes in it, then all the H+ would just kind of come back in without going through the ATPase, right? So that's why it's important to have to maintain the integrity of the inner membrane right here. Okay. Well, that has been an hour. That went by quite quickly. Let's take a quick break, 5 minutes, and then we'll continue. All right. So see you in 5.